So this is the November edition, I guess, of the COVID-19 Immunology Consortium-BC Symposium. I'm really excited for our speakers today and to hear what they're going to say. So I'm going to try and not take up too much time to give them the stage. So we have Chad Poloni and then Carlsten and Manali Mukherjee who will be presenting today. I'm just going to start with a land acknowledgement. Just to say that BC Children's Hospital Research Institute, which is where I am based and also or close by to where I live, operates on the traditional, ancestral and unceded territory of the Coast Salish peoples, including the Squamish, Musqueam and Tsleil-Waututh nations. And this acknowledgement, gratitude and respect extends to all of the First Nations communities on whose traditional territories that we build. And I'd urge you to all take a moment to think about the territory that you are on, wherever you are today, and that we're here as guests of the First Nations of these lands. So just want to, for people who are sort of new to this group, just to briefly just mention what the purpose of this consortium was. So it's an initiative that's funded by the UBC Faculty of Medicine as part of the work that they're doing with COVID-19. And the goal is really to bring together researchers at all stages of their career to help facilitate collaborations with individuals who are interested in infectious disease immunology, which may be people who are doing immunology related to or unrelated to infectious disease who have now sort of entered this COVID world, or people who are not necessarily doing the immunology themselves, but have studies which may be enhanced by additional immunologic work. And so right now we have over 100 researchers across BC who are part of the consortium, which is really exciting. We're going to try and be a bit better about sort of a bit more communication and quarterly newsletters and these research symposia every couple of months. And we have a very new and energetic, enthusiastic student and trainee group. And so I guess just if people have any ideas for what they would like to see or hear in a future symposia, please let me know, and then we can definitely look at that. And if you're a student or a trainee and would like to get involved in that group and you're not already, then again, let us know and that we can try and facilitate that. So today's presenters, as said, we have Chad Poloni, Dr. Carlsten, and Dr. Manali Mukherjee. So I think without any further ado, we'll get into things. And so the first talk, and Chad, I'll just stop sharing my screen, and so I'll let you share your screen so you can drive your own slides while I introduce you. The first speaker is Chad Poloni. So he graduated from McGill University with a master's in microbiology immunology, studying chronic CMV infection, inflammation, and aging. He started his PhD in Ted Steiner's lab last fall, and his current research focuses on investigating type 1 regulatory T cells as therapy for inflammatory bowel disease. He's also developing T cell assays to identify food allergens in pediatric eosinophilic esophagitis patients and using antigen-specific T cell assays to track COVID-19 vaccine responses in various groups. And that is what he's going to talk to us about today. So Chad, over to you. And your slides.
Okay, can you all see the full screen now?
Yeah, looks great.
Okay, sweet. So my name is Chad Poloni. I'm a PhD student in the Steiner lab, and today I'm going to be talking about COVID-19 vaccine T cell responses. Okay, so just to give a current overview of the current studies we're involved in at the BCCHR. First, we have the uppercase prevent study, which is led by Dr. Manish Sadarangani. Some definitions for the study is V1 is the day of the COVID-19 vaccine. V2 is 1 month post-dose 1. V3 is at time of dose 2. V4 is 1 month series completion or after dose 2. V5 is 4 months post-series completion. V6, 7 months. V7, 10 months. V8, 13. V9, 19. So we have a total of N 51 AIM assays in ex vivo phenotyping, which were all run on fresh blood. I'll go in more detail about the AIM assay later. This study includes a total of 18 individuals, and today I'm going to be presenting preliminary data from an N of 4 individuals. So we have data from visits 1, 2, 4, and 5. The next study is the lowercase prevent COVID study, which is looking at vaccine responses in transplant patients. These have all been collected as PBMCs so far, and we have not run any assays on them, but the assays will be done on PBMCs. We have a blood cancer study, which is looking at vaccine responses in blood cancer patients. We've done a total of 30 AIM assays on fresh blood, and we also have 220 PBMCs collected. And lastly, we have a solid tumor cohort, and half of this cohort is made up of a group that's receiving traditional chemotherapy, and the other half is receiving other biologics. And all of these studies are undergoing follow-up visits, so none of them have been concluded yet. But I think all of them we've finished enrollment. So an overview of how we do our sample processing on the fresh blood. First, we are doing ex vivo phenotyping. We do surface staining and intracellular staining for various T cell markers, such as Tregs, naive cells, memory cells. And then this is all run within 24 hours on a flow cytometer. In parallel, we perform an AIM assay, which we take Buffy coat after spinning down the fresh blood and taking enriched PBMCs, which we mix in a 1-to-1 ratio with an IMDM media, which does not contain FBS to prevent any background stimulation. These are incubated with various stimuli. And then for 48 to 48 hours, we can then stain these cells and measure them with a flow cytometer. And then we are also freezing PBMCs in plasma if we want to go back later and perform any additional assays. So to go more into the antigen-induced marker assay, or the AIM assay, this assay allows us to detect antigen-specific cells by just looking at surface markers and using flow cytometry. So first, we'll take 100 microliters of Buffy coat with 100 microliters of IMGM and plate this with various antigens in a 48-well plate. For the COVID study, we have an unstimulated control, the S peptide, the N peptide, and the N peptide. For those receiving the vaccine, we only expect to see a response to the S peptide. If we see a response to the M or the N peptide, we can indicate that they had a suspected COVID infection during vaccination or after vaccination. We also have agriflu, which acts as an antigen-specific positive control. So anyone that's received a flu vaccine should have a response to that. And then we have cytostim, which is a non-specific positive control, which basically cross-links to TCR and causes activation. This last control is very important in our immunocompromised cohorts because some of these patients don't have any response to cytostim at all. So so then they wouldn't have any response at all to any of these antigen-specific stimuli. So these plates are incubated for 48 to 48 hours at 37C. And then we stain them for CD4 or CD8 along with activation markers. In the case of CD4, we're looking at the upregulation of CD25 and OX40. And in the case of CD8, we're looking at the upregulation of CD69 and CD137. And we can measure these readouts with a flow cytometer. So in the case of CD4 positive antigen-specific T cells with the that are specific to the S peptide, we have detection of the S peptide to the TCR, which causes signal transduction and upregulation of IL-2 receptor alpha. And then we have autocrine signaling through the CD25 receptor in combination with signaling through the TCR that causes the upregulation of OX40. And this peaks around 44 hours, and we can detect it up to 48 hours. So in combination of these two receptors, we can detect antigen-specific T cells. And just to give you an example of what these flow plots look like, we have the unstimulated here. On the y-axis, we have CD25, and on the x-axis, we have OX40, and we're looking at this quadrant 2. And then we have the same patient incubated with S peptide, and you can see a nice increase in both CD25 and OX40, indicating the antigen-specific T cells here. And just a caveat, in the beginning of these studies, we're using peptides from Miltenyi, and they only had the S-prot available, which was the predicted immunodominant sequences. However, they later came out with the complete S peptide, which contains all of the amino acids. So we started to add this into our assay halfway through visit 2s and then all visit 3s and onwards. This complete S does give better responses in the CD4s. However, we don't seem to notice a difference in the CD8s between the two. So now to get into some preliminary responses for the CD4 T cell responses. We can see at time point 1 baseline, all of them are below our cutoff threshold, which is 0.02%. At visit 2, which is 1 month post-dose 2, we see 2 individuals slightly above that threshold. And then we see a peak for all individuals at 1 month post-dose 2, with the exception of ID12, which seems to peak even higher at visit 5, which I'll talk about in the next slide. What's interesting also is our agriflu, or our antigen-specific positive control, follows a similar pattern to this S peptide. So what we think is happening is somehow the mRNA vaccine is priming or acting as an adjuvant to allow other antigen-specific T cells to be more responsive once we take them out of the body and stimulate them as well. We don't see as clear of a pattern in our non-specific cytostim control. However, we do see that with this one individual with 8. So we'll need to increase the N. So once we get more individuals at visit 5, it will be interesting to see if this pattern holds true. So going back to this ID number 12, we can look at their response to the N peptide and to the N peptide, and see that both of these responses are above, for the N, well above the threshold, indicating that they did have a COVID infection somewhere between visit 4 and visit 5, which caused the response to increase further. Okay, so now moving into the CD8 responses, the magnitude is much less than the CD4 responses. However, at time point 4, we can detect 2 individuals with a positive response, and we see that patient that had a probable COVID infection have a positive response after visit 5 as well. The agriflu doesn't seem to follow that similar response, with the exception of ID8, which has a very strong peak at visit 4. However, cytostim does seem to follow that pattern again that we saw with the CD4s and the agriflu, which is interesting. So just a summary of what we're seeing is that antigen-specific S-peptide CD4 T cell responses peak at visit 4, which is 1 month after series completion. CD4 T cell responses are still detectable at time point 5, which was 4 months after series completion. ID12 has a suspected COVID-19 infection between visits 4 and visit 5, causing a peak of the response at visit 4, and this is determined by the N and the M peptide responses. Agriflu CD4 T cell responses follow a similar pattern as the S peptide. It could be due to the timing of the flu vaccination. However, this is unlikely as these responses were detected in August, or it's some priming or the single-stranded RNA vaccine from the vaccine acting as an adjuvant to kind of boost these T cells. And lastly, the magnitude of the CD8 specific T cell response is much lower than the CD4 T cells. So now moving on into the ex vivo phenotyping. This was carried out on fresh blood for all of these individuals using a 5-laser symphony. And we have 19 different markers, which allows us to look at naive cells, effector memory, central memory, follicular helper, Th1, Th2, Th17, and Th1.17. So we can do traditional Boolean gating as shown here to look at all of these different T cell subsets, or we can utilize dimensionality reduction and clustering techniques to identify novel T cell subsets. So that's what I'm going to talk about a bit more in detail today. So the 2 analyses that I used was first TISNE, or T-distributed stochastic neighbor embedding, which is a dimensionality reduction technique allowing for the visualization of high-dimensional data. And it gives each data point, or in this case, a cell, a position in 2D space, and the points are grouped by similarity. So in this example here, you see the TISNE is able to group the variety of different immune cells based on the markers, and it does that very well. Next, I use FlowSum, which is another dimensionality reduction technique that allows visualization of high-dimensional data sets. And it uses the generation of self-organizing maps of clusters based on markers, and it assigns each cluster to a metacluster, and then it shows the population abundances and marker expressions. So for our analysis pipeline, first I cleaned up the FCS files by gating out single cells and only CD3 positive cells. I then downsampled all events and IDs to 8,000 events, so we weren't getting different numbers of cells from each ID or each visit to kind of skew the data. I then concatenated all of the samples into a single FCS file and ran the t-SNE on that single FCS file. I could then go back onto that master t-SNE and overlay each individual visit to identify how the phenotype changed over time from each visit. I then also performed a flow sum of the FCS file as well. And then I could also gate each individual visit on top of the populations that were generated from the flow sum analysis. And this is just an example of one of those populations. And you can see that population is 99.2% made up of visit 2. So that's really only appearing during visit 2. I can then go back and overlay those flow sum populations on top of the t-SNE to determine the phenotype and identify any T-cell subsets. So just to give you an overview of the t-SNE analysis that was run on the ex vivo phenotyping, in the black is the master t-SNE that includes all of the visits, and then the different colors are each individual visit overlaid on top of those. So we have in the green visit 1, in the blue you have visit 2, so we have this identification of a new cluster popping up during that visit. At visit 4, which is 1 month post-series completion, we have a generation of this cluster here. And then in visit 5, we have, it's relatively unchanged. However, this cluster kind of decreases and we have a bit more concentrated in this cluster as well. So we can go back and interrogate these different clusters. So for example, in visit 4, we saw this cluster was enriched. So I can overlay various heat maps for different parameters. So for instance, this particular cluster is CD4 positive, enriched for FOXP3 and CD45RO. So it looks to be a memory Treg subset. I then also performed flow sum on all of these different visits, which generates a variety of different populations that are best suited to our data set. And then it gives me a heat map for all of our different parameters we're looking at. So for instance, these particular populations are mostly CD4, and these populations here are CD8. I can then go and look into each population to see what visit is enriched in those populations. So for example, in visit 2, it's population 19 and 6 are majority only from visit 2. These cells are both CD8 positive and CD4 positive, and they're both CD45RO positive and CTLA4 positive. Visit 4, we see an increase in Tregs. So we see CD4 high, Helios high, CD39 high, and FOXP3 high. And then in visit 5, we see majority CD8s that are CD45RA positive and CCR7 negative, so potentially a TEMRA cell subset. So these cells are developed after continuous antigen stimulation. So I can then go back and overlay these flow sum populations onto my original t-SNE to better interrogate their phenotype. So for example, at visit 2, we saw was enriched during population 19. I can see that these cells are majority CD8 positive, CD45RO positive, and CCR7 low. So it looks like to be generation of effector memory CD8 T cells at this time point. I can do the same thing for visit 4, which again was enriched at population 5 for CD8s. For CD4s, Helios and FOXP3. And we also see an increase in CCR4. So this is maybe a potential CD4 Treg humoral response occurring at visit 4. And finally, at visit 5, I can see an increase in CD8s that are both CCR7 positive and negative. So we have an increase in both naive and TEMRA CD8 T cells. So a summary is that t-SNE and flow sum allow for dimensionality reduction and overview of all of our phenotypic data, visualization of all parameters to identify T cell subsets that could be missed during traditional Boolean gating. At V2, we have identification of CD8 T cell expansion. At V4, we have identification of a potential Treg subset and also a humoral response occurring. At V5, we have resolution of the initial vaccine response. A majority of these cells are naive CD8 T cells, and we also have that identification of the TEMRA CD8 T cells as well. It's important to note that all of these subsets need to be confirmed with traditional Boolean gating, so we can run stats and include clinical data as well. So we'll do that once we get more N's into our visit 5s. So our future directions is to generate t-SNE and flow some analyses at all time points to confirm the populations using Boolean gates, and then to go back and correlate to see if these subsets can predict T-cell responses. So for instance, if we have a large increase in Tregs at V4, does this dampen the T-cell response at V5? And then also to compare immunocompromised patient cohorts to the prevent cohort. So I'd just like to acknowledge all of the labs that are leading all of these different studies. So the Levings lab, the Siddharangani lab, Dr. Stephen Chia, Dr. Souther Heatherland, and Dr. Sarah Belga. So thank you for looping in our lab to do some of the T-cell work with you guys. And thank you for the opportunity to speak here today.
Thanks, Chad. Thanks, James. So it gives me a great pleasure to introduce the next two speakers. So I believe it's going to be a tag team effort. So I'll let you get your slides up while I do the introduction. So Dr. Chris Carlsten holds the AstraZeneca chair and is the Canada Research Chair in Occupational and Environmental Lung Disease. And he is the director of the Air Pollution Exposure Laboratory and the Legacy for Airway Health, the theme lead for personalized health in the UBC Faculty of Medicine, and also holds positions at the Peter Wall Institute for Advanced Studies, the UBC School of Population and Public Health, and Center for Heart and Lung Innovation. As a clinician scientist, he focuses on how air pollution affects our lungs and immune system in a public health context, and he leads a team of healthcare professionals, researchers, and trainees conducting human exposure studies to common air pollutants such as diesel exhaust. And they use clinical and laboratory techniques to understand how air pollution exposures impact lung diseases and leverage this knowledge via integrated knowledge translation and stakeholder engagement to develop approaches that reduce health impacts in Canada and beyond. I guess of relevance to what he's going to talk about today has been leading, looking after people after COVID-19 and sort of post-COVID implications. The second speaker is Dr. Manali Mukherjee. So she completed her PhD in airway cell biology from the University of Nottingham in the UK. And after a short stint of research at the National University of Singapore in clinical autoimmunity, was a postdoc at the Firestone Institute for Respiratory Research to investigate autoimmunity in eosinophilic asthma. She stayed on there to establish a lab at McMaster, focusing on lung autoimmunity and biomarkers. Her work on investigating novel autoimmune mechanisms in complex airways disease and of recent COVID is supported by multiple sources, including CHR, and she's the current recipient of the CHR Emerging Researcher Award in Asthma and has over 40 publications in top journals in the past few years. And she also is interested in understanding autoimmunity in COVID long haulers, supported by CHR and the COVID-19 Immunity Task Force. So I will hand over to both of you, and thank you again for joining us and sharing your work today.
Cool. Thanks, Manish. We're just by our prior arrangement, I'm going to start. And I'm going to also start by saying that as I get farther in my career, I get smarter and smarter about teaming up with people like Manali. So she was kind enough on relatively short notice to join in, and we both agreed ahead of time that we are going to explain conclusively every single thing you see on this slide, and it's all going to be related to the data that Manali shows. So you may not need any more of these meetings because it'll all be solved. But seriously, we do think there's some real impressive preliminary data to share, and this is a example of real interdisciplinarity. You'll see I'm focusing on more clinical aspects, but Manali's work, I think, is from an immunology perspective really got some potential explanations. So, okay, so this is what happens sometimes when I try to, I'm going to go to my other, thankfully I learned from the past that I'm prepared for this. If I try to run it right off of my kind of online SharePoint file, it doesn't like it, but here we go. So you guys can see that, right? I'm assuming I just switched over.
No, we can't see anything, Chris.
Oh, okay. Interesting. Let's try it again. let's try it again. Let's make sure I get the right one. Okay, I think this is it. Now let's go full screen. So now you see the virus.
Yep. Yes.
Okay.
On a timeline.
Perfect. Thanks. Yeah, it's funny how you can't do it right off SharePoint. But anyways, I guess this probably doesn't move fast enough on the Ethernet. The timeline here is really just to set the stage for the clinical aspect, in the sense that a year, more than a year ago, obviously, a year and a half now, April 2020, we came together, a few of us, and said, you know, we got to jump on this. And I remember this time I was on my bike and pulled over on the side of this nice little road in Delta and had this 2-hour conversation with my colleagues. Anyway, we set up this clinic that's now expanded. I'll show you. But it was relatively record time. We, from the very beginning of the conversation, it was about a month. And those are the patients that you'll be seeing data from. So it's also a tribute to the patients that quickly got on board, and a whole process where we were very, we believe it modestly kind of state of the art in terms of the engagement with the patients. And that's kind of just gotten even better, I'd argue, but not, you know, huge numbers at the beginning. And obviously we saw what happened, and we used basically the last 18 months to get this network up to speed. And some of you guys know about it. But today's going to be one of the, I think, best examples of payoff, I guess, from all of this. So early on, we started with respiratory outcomes. As you can imagine, that's kind of my field, and it's logical, of course, with COVID. And I'll show you just briefly a few studies, very kind of fundamental, not in the basic science sense, but in this is what happens to people's lungs from an imaging and functional perspective. So more than half had a pulmonary function abnormality on very basic clinical. These are not esoteric tests. These are the same tests that we do for patients in routine day-to-day practice. So a very common finding, which, you know, in the general population, if you just pulled people off the street, this would be maybe 5%. So massive problem right there. And then CT scanning showing, again, more than half that had an abnormality that we consider for sure clinically significant. I always tell people, if someone said, oh, I'm just going to, people say 10%, that's not that great. Well, if someone said, I'm going to take 10% of your lung and scar it, you probably wouldn't be super happy about it. So these are, and the reticulation, which is a sort of a technical word for scarring, is generally irreversible. So these are not trivial things. But then, of course, we want to know from the patient perspective. So looking at patient-reported outcomes is what this study looked at. And what this shows and was and remains disturbing is that about 2/3 of these patients had an abnormal reported outcome. And these are, again, not esoteric things. These are basic quality of life, shortness of breath. You can see mood. And the key thing here is that the general population normal range are the gray bars. So this isn't just the average, this is the whole range. So the general population should sit, all the general population within this gray bar, but you see all these people that are falling outside of it. So even though obviously we didn't test these people before they got COVID because we had no idea this was coming, really, you can't just say, oh, this is just a reflection of their background, because it would be completely illogical to do so. So this is true abnormality, as was the CT scanning and pulmonary function data. Then we started looking at changes, and we were seeing these people a lot. At least every few months, and they gave their valuable time. But in the first 3 months, we were relatively encouraged. On the bottom, you can see pulmonary function improvements. They're not massive in terms of quantifying, but they're still improvements nonetheless. The quality of life improved not as much, which is an important point here. It's nice to have a modest improvement in your lung function, getting closer to the normal range, but they're still complaining of these basic symptoms. On the dyspnea or shortness of breath front, we started getting into this idea that there's what we call explained and unexplained dyspnea, and it's a crude way to describe it, but it basically means that if your UCSD, which is a self-reported score for shortness of breath, is high, and your DLCO, which is diffusion, it's kind of the most fundamental metric in the lung, which is basically gas exchange. So if your gas exchange is good, but you're still complaining of shortness of breath, well, it's unexplained essentially. It's more complicated than that, but it's a reasonable way to think about it. And as you can see at the top right, 18% of all the people we followed had this unexplained category. And just of the people that complained of shortness of breath, because thankfully not everyone post-COVID has shortness of breath, but of all those post-COVID that did have shortness of breath, about half of them were unexplained. So there's something happening here that we needed to dig into further. And another problem was that by 12 months, so we just finished these data a couple months ago, by 12 months, they're starting to plateau in terms of their improvement, as you can see here quite consistently, and that's unfortunate. Thankfully, most of them are getting closer to normal, but not totally normal. So we're seeing this kind of plateau and improvement. And the same thing with the symptoms or the self-reported outcomes. So the one that had been the most kind of encouraging early on, you can see at the bottom this initial improvement over the 3 to 6 month phase is now flat out unchanged in the subsequent 6 months. So not fun for those people that are still suffering from that. So what's going on? Obviously, there are lots of hypotheses, but we put this network together with lots of support, huge effort to get all kinds of government and non-government and health authority, et cetera, entities on board. It's an ongoing struggle, but we've made some major breakthroughs finally on that front. And it's all because, as you can see from this one JAMA paper, it's outdated, but it's essentially unchanged in that the post-COVID period is marked by primarily fatigue and dyspnea. And of course, there are a lot of other symptoms, but you see that this kind of cluster that was obvious in the acute phase largely tapers off, thankfully. But the two things that don't, or the two that taper off the least, or the most persistent, are fatigue and dyspnea. And fatigue especially, I think, is something that is incredibly vexing and frustrating to patients, especially when people don't take them seriously. But there's obviously something physiologic going on that we're trying to understand. So this is the network. I'm not really here to, I guess, cheerlead this, but you should be aware of it. It's a pretty big deal for the province in the sense that this is the kind of network that people have been advocating for kind of in the background for decades on different ways regarding chronic disease, and it never happened because it wasn't the crisis known as COVID. And if there's anything good that came out of COVID, it's that it was enough of a crisis that it established this network that we think is going to persist well beyond COVID and be a model for other chronic disease if we can maintain funding. Part of that network is a biobank, and that's going to lead to my handoff to Manali in a second. But we were able to get blood from almost all these patients. Again, I may not have made it totally clear, but the network was founded initially on people who are discharged from hospital, because at least at first we thought they were the ones who needed the most attention. Of course, now we know that people who are never hospitalized have all kinds of problems that need to be addressed. But anyway, it started out with post-hospitalization, and we get a rich set of those blood samples that were used for several projects. But again, the one I'm excited about in a kind of scary way, as you'll see, is the one that Manali has been working on with us.
Thank you so much, Chad and Chris and Manali, for sharing the data. I'm sure you could all have spent twice as longer, or if not even longer than that, going through the data. I'm sure some of us would love to do that. So maybe if people have specific issues, then we can reach out separately to you guys. And I just want to also huge thank to James for helping organize this and coordinate this, and to Beth White, who you haven't seen or heard from, but in the background has basically been organizing the entire thing. So thank you so much, Beth, and to the whole steering committee of the consortium. With that, I will call it to an end, and thank you so much, and enjoy the rest of your day and have a good weekend, everyone. Thanks.